Covid-19. January 14th. Daylight. The virus has overtaken the body. Somehow, I'm still alive. Thanks to a sneeze by an infected patient, COVID-19 was able to enter the body through the nasal cavity. The main targets and the primary site of infection for COVID-19 were the lungs. After the successful entrance of the virus through the nasal cavity, the virus went all the way to the alveoli, going through the larynx, trachea, primary, secondary, and tertiary bronchi. When the virus reached the alveolus, it was looking for a specific receptor to bind to. This receptor is referred to as ACE2, or angiotensin converting enzyme 2. Unfortunately, the receptor was expressed on the surface of one of the cells in the alveolus. The cell is called type 2 pneumocyte. The virus attached itself to the cell. It entered the cell through endocytosis. It was able to copy its RNA, proteins, and enzymes, and therefore it was able to make copies of itself. After the virus made hundreds of copies of itself, I, the alveolar macrophage, decided to investigate what's going on in the alveolus. So I approached one of these viruses and I engulfed him after recognizing him through one of my PAMs, also known as pathogen-associated molecular pattern. After the phagocytosis, I realized that the situation is much more worse than I first expected. So I decided to release some of my cytokines and call for my brethrens from the innate immune system. The first response came from my brother, dendritic cell. We held long conversations about how exactly are we going to deal with these viruses. He had his own plans of taking it to the adaptive immune system, but I disagreed at first because I thought if we just release more and more cytokines and induce inflammation, then the viruses are going to disappear eventually. If we just focus on phagocytosis, then everything will be fine. Unfortunately, I was wrong. But he agreed with me at first, and we decided to try the cytokine plan. The excessive production of cytokines did not work out. It caused increased production of mucus and constricted airways, which decreased significantly respiration. The body was about to die at this point. So the dendritic cell, and rightly so, decided to take the matter to the adaptive immune system. The dendritic cell engulfed one of the viruses after it recognized it through one of its spams and digested the virus through phagocytosis. The cell then traveled to one of the secondary lymphoid organs, the spleen. After reaching the spleen, the dendritic cell looked for a specific type of T cells, the ones that can bind to the antigens obtained from Covid-19. After finding them, the dendritic cell decided to negotiate the possibility of activating them. After a long conversation with the T helper cell, the dendritic cell decided to present an antigen obtained from the virus to both the T helper cell and the T cytotoxic cell. The dendritic cell presented the antigen to the T helper cell through the major histocompatibility 2. After the connection, the dendritic cell produced some cytokines to activate the T helper cell. The same process happened during the activation of T cytotoxic cell, except the activation happened through the major histocompatibility class 1. The further activation of the T cytotoxic cell needed cytokines from the T helper cell as well. Both the T cytotoxic cell and the T helper cell proliferated after activation. At the same time, a B cell was able to catch one of the viruses through its B cell receptor. The receptor was matching one of the antigens found on the virus. The B cell ingested and digested the virus and presented one of its antigens on the surface through the major histocompatibility class 2. The previously activated T helper cell decided to activate this B cell by binding to the antigen and producing cytokines, which helped activating the B cell. After activation, the B cells decided to proliferate and switch the class of the antibodies on their surfaces to ones that might suit the coronavirus much better. These B cells then started shooting their antibodies, hoping that this can stop the infection. All of the antibodies produced were able to attach themselves to the viruses. As a result, the virus was not able to infect any cell anymore. In fact, the immune system was able to get rid of the viruses in a much faster rate than before through different processes. One of them, and the most effective one, was through the ADCC, also known as the antibody-dependent cellular cytotoxicity, which was carried out mainly by our friend, natural killer. At that point, the infected cells inside the body were able to capture some of the viruses produced inside the cells. The viruses are digested through a structure referred to as the proteasome. The antigens resulted are then presented through the major histocompatibility class 1. This presentation was like a red flag for the T cytotoxic cells. As a result, the T cytotoxic cells were able to identify the infected cells and activate apoptosis in every single one of them. The infected cells were all killed. The body returned to its normal status. The alveoli are all clear. The cells are all clear of any viral activities. The lungs are all healthy again. No inflammation, no infection. And we're still here and alive. So this concludes our video for today. If you like the video and the content of this video and this channel, then please like, share, and subscribe. You may also visit our website for further updates in the future. Thank you very much, and see you in future videos.